Now let's get into the mechanisms of cell division. First, a quick overview of the process. We begin with a parent cell. Remember that each cell has two of each type of chromosome. In this case, there are two big ones and two smaller ones, a red and a blue of each, or two homologous pairs. Pause and think about this for a moment. It's the language of chromosomes that is the toughest part of understanding mitosis and meiosis. Now, we agree that each daughter cell needs to end up with the same number of chromosomes as the parent cell, right? So in order for that to happen, we need a copy of each chromosome, or to make a copy of the cell's DNA. This, you recall, happens during S phase of the cell cycle. Let's review the language of chromosomes in detail, because this is the part that trips most people up. After the chromosomes are copied, each chromosome is composed of two sister chromatids. Agreed? And thus each homologous pair is made of two pairs of sister chromatids. It's the language again. Pause and get clear. Even rewind to hear it again if you need to. Okay, a closer look. Here's a duplicated chromosome. It's one of a homologous pair. It was duplicated during S phase of the cell cycle. Each sister chromatid is held to its exact copy at the centromere. If we were to add the other of the homologs here, it would look sort of like this. Now technically, for each chromosome, there are 4 chromatids, each in 2 pairs. This is also known as a tetrad. Now keep in mind that as soon as the sister chromatids separate during mitosis, they become individual chromosomes and will end up in separate cells. It's a language thing, but it's really important. So again, take a moment to think it through. Pause and rewind if you need to. Moving back out to the whole cell, when an organism has 2 of each chromosome in its cells, those cells are considered to be diploid, or 2N. This is where N equals the number of different chromosomes, which in our case as humans is 23. And the 2 tells us how many sets of chromosomes there are. So humans have 2 sets of 23 different chromosomes, or 2 of each of 23 chromosomes, where 2 times 23 equals a total of 46 chromosomes. In this cell, 2n equals 4, because there are 2 of each of 2 different chromosomes, a red one and a blue one. As we'll see later in the course, some organisms have more than 2 sets of each chromosome. So they could be 3n or 4n. So that means either 3 or 4 sets of chromosomes, or even more. Mitosis, the parent cells are 2n, and because the chromosomes were copied and then split, each daughter cell is identical to the parent cell, containing 2 whole sets of chromosomes, also 2n. The next thing we need to think about is how the chromosomes separate. This process involves the spindle apparatus. There are 2 types of spindle fibers that originate from the centrosomes of the cell's poles. There are kinetochore fibers that literally rip the sister chromatids apart from each other. And there are polar fibers that push on each other to elongate the cell and push the poles apart.
An adult human contains an estimated 100 trillion cells, and yet we start life as a single cell. To grow, develop, and repair tissue damage, we rely on cell division. In eukaryotic cells, this process is accomplished by a series of well-orchestrated steps called mitosis. Every day, our bodies must produce millions of skin cells to replace those lost through normal activity. Each of these cells must have a complete complement of the genetic material. Prior to cell division, where the cell divides into two identical cells, the DNA needs to be replicated so that each daughter cell receives an exact copy. Following DNA replication, the chromosomes condense in the nucleus of the cell. DNA condenses by wrapping around cores of histone proteins, forming nucleosomes. This beads on a string structure is called chromatin. As a cell prepares to divide, chromatin coils up further, shortening and condensing the chromosome. The replicated chromosomes are called sister chromatids. The replication of DNA and the formation of sister chromatids is one part of the entire cell cycle. To prepare for cell division, the cell goes through interphase, which can be divided into three distinct phases. During G1 or gap1 phase, all the organelles and cytoplasmic components, including the centrioles in animal cells, replicate. Then during S or synthesis phase, the DNA replicates. Finally, during G2 or gap2 phase, all the enzymes needed to aid in the process of cell division are produced. Most eukaryotic cells spend a great deal of time in interphase, and a very short period of time actually dividing, a process called mitosis. The cell is now ready to go through mitosis, which consists of prophase, metaphase, anaphase, and telophase. During prophase, the chromosomes condense and become visible, appearing as two sister chromatids held together at the centromere. The cytoskeleton disassembles as the spindle begins to form. In animal cells, centrioles play an important role in the distribution of the chromosomes in the dividing cell. The centrioles migrate to opposite poles, establishing a bridge of microtubules called the spindle apparatus, and the nuclear envelope breaks down. Towards the end of prophase, chromosomes attach, by proteins in their centromeres called kinetochores, to microtubules from each pole, moving the chromosomes toward the equator of the cell. During metaphase, all chromosomes are aligned at the equator of the cell, called the metaphase plate. Anaphase begins with the degradation of proteins that hold sister chromatids together, freeing individual chromosomes. The free chromosomes are then pulled by their kinetochores to opposite poles. At telophase, a cleavage furrow forms in the center of the cell. This indentation is made from a constricting belt of actin filaments surrounding the inside of the cell's circumference. Chromosomes cluster at opposite poles and begin decondensing as the nuclear envelope reforms around them. The spindle apparatus disassembles as the microtubules are broken down into tubulin monomers that can be used to form the cytoskeleton of the daughter cells. In animal cells, cytokinesis completes cell division by extending the cleavage furrow to completely separate the newly formed daughter cells. Since plant cell walls cannot be constricted by actin fibers, vesicles form an expanding membrane partition called the cell plate. Like animal cells, plant cells use cytokinesis to finish the division of the contents of the cytoplasm between the two identical daughter cells.
As you just saw, mitosis can be divided into a series of phases. Prophase, where the chromosomes are assembling themselves and the nuclear envelope is breaking apart. Metaphase, where the chromosomes line up single file down the center of the cell. Meta means middle, so this phase is pretty easy to remember because the chromosomes are right down the middle of the cell, and it's in the middle of mitosis. Anaphase begins at the moment the sister chromatids are pulled apart and become daughter chromosomes. And during telophase, the daughter cells begin to form, and new nuclear envelopes form around each group of chromosomes. there's cytokinesis, where the cell itself is split in two to form two new daughter cells that are identical to the parent cell. Although mitosis happens spontaneously in our bodies and is controlled by specific genes, I call them the gas pedal and brake pedal genes, we can also create environments in the lab that favor cell division and the growth of specific tissues. I'm sure you've heard of stem cells, but why are they so interesting? They're interesting because they could become any type of tissue depending on the signals they get from their surrounding cells, which means that we have the potential to cure human ills by regrowing, say, nerve cells to treat spinal cord damage, or pancreatic cells to help those with juvenile onset diabetes. Saving cord blood is a great way to keep your child's stem cells around. You never know. They may be useful in the future, either for the child or for genetically similar family members. Something to think about. Now, we started out talking about cancer and cell division, but let's take a moment to consider it again. As we've learned, it's uncontrolled cell division that results in undifferentiated, essentially immature cells, tissues that do not grow to behave like the surrounding tissue. And in the end, they impede the proper function of that tissue. But how does it happen? Well, there are several modulating genes that make proteins involved in controlling the rate at which cells move through the cell cycle that we looked at earlier. These are the gas pedal and brake pedal genes that I was talking about. If the gas pedal gets stuck, we're in the go position. Or if the brake pedal doesn't work, then the cell can't slow down and is dividing out of control. We'll take a look at a few of the major cell cycle control checkpoints. There is one here in G1. It's especially important because this is where there are mechanisms to check that the chromosomes or the DNA was copied and separated properly during the last division. If the DNA has become damaged, then a regulatory protein called P53 tries to repair it. If it's beyond repair, P53 will act to stop the cell cycle from progressing and cause the cell to die, something that we call apoptosis, or cell death. Many cancers seem to have a damaged P53 regulatory protein. So in essence, in this case, the gas pedal is stuck in the push-down position. The G2 checkpoint involves another set of regulatory proteins that check to see if the DNA was replicated properly during S phase before we go into cell division. Again, the cell will not be permitted to enter mitosis if something's wrong. Another key checkpoint is the M checkpoint. Here, the chromosomal alignment during metaphase is checked. If the spindle apparatus fails to arrange the chromosomes correctly, mitosis will not continue. Now, these are all really cool control mechanisms that work together to ensure things operate as they should. But sometimes these cell cycle control genes become damaged, or as we call it, have mutations. These mutations can be caused by any number of different agents, like radiation, or smoking, or bad diet, and a variety of environmental toxins, or as we're learning, even viruses can bring on cancer. In the images on the right, you can see the progression of cervical cells affected by HPV, the human papillomavirus. And they're progressing into cancerous cells in the bottom image. Cancer cells tend to never differentiate into the tissues that they're embedded in and usually have abnormal looking nuclei. You can see the difference here from the normal on the top to cancerous at the bottom. Because cell division is out of control, these cells form tumors that also secrete chemicals that tempt blood vessels to grow in their direction in order to provide them the nutrition they need to grow and divide and produce more cells, thus growing a tumor. This process is called angiogenesis. Metastasis occurs when the tumor gets into that blood supply that was formed during angiogenesis and land in other places in the body to form other tumors. So it's really our individual responsibility to do what we can to protect ourselves. Smoking's one of the biggest factors in creating cancer, and not just for lung cancer and throat cancer. Diet's also a huge consideration. This is a saying that I love. If it's a plant, grows on a plant, or eats plants, then go ahead and eat it. But if it's made in a plant, you should probably leave it alone, or at least limit your intake of it. Really, this is a topic of a whole other course, but certainly something we should pay attention to. To finish up this section, let's take a quick review of the cell cycle controls.
During the cell cycle, certain checkpoints are encountered to make sure the process is occurring accurately. And if it is not, the cell cycle will stop at the checkpoint and correct, or possibly inhibit that cell from dividing. The first checkpoint is the G1s checkpoint, and is considered the primary point at which the cell cycle continues or stops. External signals and growth factors can influence the cell cycle and affect the progress at or before this critical checkpoint. The G2M checkpoint allows cells that have successfully completed all 3 phases of interphase to begin mitosis. The last checkpoint is the spindle checkpoint, ensuring that all chromosomes have attached to the spindle in preparation for anaphase. Growth factors, the size of the cell, and the nutritional state of the cell are all contributing factors in cell cycle regulation, ensuring that only certain cells divide at appropriate times. Once all the checkpoints in interphase are cleared, mitosis can occur. From interphase to cytokinesis, the entire process of cell division can take on average 10 to 20 hours in a typical plant or animal cell. Depending on the nature and use of the cell, the process can happen at different frequencies as well. In humans, our skin cells have a high turnover rate due to wear and tear, and go through mitosis very frequently, whereas other cells, such as adult neurons and muscle cells, rarely divide. The accuracy of mitosis, as well as the consistency of the checkpoints during interphase, ensure that most cells in a eukaryotic organism can produce identical copies of themselves. This process allows for growth and repair to prolong overall physiology as well as life itself.